Now, as we venture into our discussions on genetic engineering and how we manipulate DNA to create changes or differences in organisms, we need to remember that we're not just working with DNA, but with the DNA products. And it was in 1945 that George Beadle and Edward Tatum clearly demonstrated that proteins that are essentially the products of DNA, and it's the proteins that we're trying to actually produce, many of those proteins are going to be enzymes. And those enzymes are going to be important in different kinds of metabolic pathways or different types of biochemistry. So ultimately, as we're manipulating these various pieces of DNA, we really want eventually to produce unique proteins that can be used in all kinds of technologies, industry, manufacturing, medical treatments for diseases, and it all stems back to those early experiments performed using the fungi Neurospora and analyzing different metabolic pathways and the unique enzymes used in those pathways. Now, what are some of the things that we can do with DNA? If we simply take a look at the physical chemical properties of DNA, that's the heart, or essentially the root, of how we're going to be able to manipulate, and this is how nature has been doing it for billions of years, the DNA or hereditary material inside of an organism. As it turns out, when you heat different kinds of biochemicals, like proteins, let's say, if you heat a protein beyond its temperature maximum, that protein is essentially cooked, or what we call denatured. And once a protein is denatured, it can no longer be cooled and reversed to its original characteristics. That's very different, or distinctly different, than what happens if DNA is denatured. If you heat DNA above its temperature maximum, you'll notice in the illustration on this slide that the two sides of the DNA ladder separate because the hydrogen bonds are broken with extreme heat. The heat breaks the hydrogen bonds, but notice that the sugar phosphate backbone, the red and the blue, two opposite sides of the DNA ladder, are still intact. Those phosphodiester bonds are much tougher than that. You'd have to heat much higher temperatures or be exposed to much higher sources of radiation to break the phosphodiester bonds. So typically when we elevate the temperature above the temperature maximum, not too terribly high, but beyond the temperature maximum, the two sides of the DNA ladder separate. So we have single-stranded DNA, but the two opposite sides of the ladder stay intact. So we have two complementary, which means nucleotide pairs could be formed on both sides of the ladder. And so if we denature the DNA with heat, again, just high enough to break hydrogen bonds, but not phosphodiester bonds, and then cool, the two sides of the ladder go back together the way they were. Now, in addition to temperature, pH can also cause proteins to denature and nucleic acids to denature. Usually when we talk about denaturing, we think of temperature, but acids and alkalis can do that too. So again, separating the two sides of double-stranded DNA to create single-stranded DNA, and then cooling to allow for what we call reannealing, or the two sides of the ladder to come back together in the appropriate sequence. Now, what if, for example, on the right side of this slide, what if we have double-stranded DNA from two different genetic sources? So we have red and blue and yellow and green. If we denature those two double-stranded pieces of DNA, separate them into single-stranded DNA fragments, mix all that together, all those pieces, the red, the blue, the green, and the yellow, in a test tube, and then lower the temperature, or allow the pH to go back to a more functional, normal pH. Notice that some of the reannealing will allow red-green combinations and yellow-blue combinations. And the more homology or similarity between the two sources of DNA, the more intact or complete the double-strandeds are. The double-stranded DNA on the red-green, notice it has exactly complete homology. Every single nucleotide pair matches up. Same thing's true of the yellow-blue. With that kind of information, we could essentially say the DNA from bacterium A and the DNA from bacterium B are exactly the same, or from the same species. Now, the greater the homology, the more related or the more similar the two sources of DNA. If there is very little homology, very small pieces, or just hardly any annealing or reannealing occurring, then we can say that these pieces of DNA are unrelated or very distantly related. This very fundamental physical-chemical property of DNA has become a very useful tool in a variety of new technologies using and manipulating DNA. Now, if we've taken two pieces of single-stranded DNA, they could be from the same source or they could be from a different source, but they're pieces that are single-stranded, and we allow them to anneal to form new double-stranded DNA. That process is called DNA hybridization. Now, when we talk about a hybrid, we're talking about a product from two different starting materials. And this could clearly be the case, or clearly be the example on the right-hand side of the slide, where we have two different sources of DNA from bacterium A and bacterium B that we've denatured, producing single-stranded DNA that we then form hybrids of double-stranded DNA from individual single strands of DNA from each different organism. So DNA hybrids, single-stranded DNA allowed to anneal or hydrogen bond to form double-stranded DNA. And again, the two single strands could be from two different sources. There are two types of transport devices that we use now in modern biotechnology or genetic engineering. Often we can use viruses, or we can also use plasmids. Now, hopefully viruses are pretty straightforward. These are the acellular microorganisms that we typically associate with infections or diseases. But viruses can be used to package bits and pieces of genetic material inside the virus and essentially just be used like transport devices or trucks, where we're going to use those viruses to deliver new genetic material. We can also use plasmids, and if you recall, plasmids are tiny, small, circular rings of DNA that we can find in prokaryotic organisms, especially like bacteria. And plasmids are the extrachromosomal DNA of prokaryotes that can have additional genetic material that's unique or not found in and on the chromosome of the prokaryotic organism. Well, as it turns out, typically there are hundreds of copies of plasmids naturally occurring in a wide variety of species of bacteria, and plasmids can be easily transferred by one or two naturally occurring lateral gene transfer mechanisms from one prokaryotic cell to another. We also have found that with the use of specific kinds of enzymes that are essentially molecular scissors, we can splice new pieces of DNA into the plasmids and then transfer those recombinant DNA plasmids from one cell to another. These are all tools that we're going to need for our new molecular genetic engineering that we have been performing now in laboratories for probably about the last 20 years or more. In 1928, or thereabouts, Frederick Griffith was a microbiologist working with 2 colony morphologies of Streptococcus pneumoniae. Now, colony morphology really is important in microbiology. Those are techniques that we've asked you to perform in lab, to take a look at colonies and describe their appearance. Is the surface smooth, shiny, rough? Is the colony elevated from the surface of the agar? What's the edge or margin look like? What's the color? All of those physical characteristics of the colonies actually reflect the characteristics of the cells. So when we say there are smooth pneumococci and rough pneumococci, we're talking about colony morphology when we look at the surface of the colony. There really are some strains of Streptococcus pneumoniae that when you look at the colonies on an agar plate, they're smooth and shiny, versus the other types that we can find that are sort of dry, rough looking on the surface. Now, these distinctive colony morphological characteristics are reflecting some unique characteristics of the individual cells of Streptococcus pneumoniae. The smooth pneumococci, the reason that they're smooth and shiny in colonies is that each individual cell, or notice that they actually form pairs or diplococci, have capsules. And the capsules are kind of slippery and shiny themselves, so when you get thousands and thousands of cells together, the whole group appear to be shiny or smooth. That's in comparison to the rough pneumococci, which don't have any kind of capsule. So that when you get, again, millions of cells of rough pneumococci together in a colony, the colony is going to appear that same way, kind of dry and rough looking on the surface. Definitely not smooth and shiny. Well, Frederick Griffiths was trying to determine why, if you expose mice to the smooth pneumococci that are living or growing, you essentially wind up with dead mice. In fact, mice are exquisitely sensitive to growing, living Streptococcus pneumoniae with a smooth colony morphology. We say that the capsule, which is the unique characteristic, is a virulence factor. It appears to be involved in the sickness and mortality of these mice. He realized it was the capsule that seemed to be an integral virulence factor, because if he took the smooth strep pneumo and he basically cooked it, or heat killed the bacteria, he would wind up with living mice. No sickness, no disease, no mortality. So the heat-killed smooth pneumococci, even though the capsule was still around or still in the mixture, didn't seem to have any effect if the bacteria was dead. If you used living smooth Streptococcus pneumoniae, he wound up with lots of dead and sick mice. If he used living rough Streptococcus pneumoniae, he also wound up with lots of live, healthy mice. So definitely the capsule seemed to be involved, but there had to be something else. You had to have some other factor inside that living, smooth Streptococcus pneumoniae. For whatever reason, maybe he was just trying different combinations. One of the combinations of materials that he used on his mice was a combination of heat-killed smooth Streptococcus pneumoniae, every little bit from that heat-killed preparation, and living or growing, non-virulent, rough Streptococcus pneumoniae without capsule. So he injected both of those materials into his mouse and quickly found lots of sick and dead mice. When he did a necropsy or collected samples from these sick and dead mice, what he found were a combination of smooth Streptococcus pneumoniae with capsule and rough Streptococcus pneumoniae without, all in the same mice. He described this process as transformation. He said that the heat-killed smooth had somehow, probably with the assistance of the living rough strep pneumo, had transformed back into living smooth, virulent, or disease-causing Streptococcus pneumoniae. Now, intervening years took a little time for scientists to come back to Griffith's discovery because I think of some of the political changes, especially World War II, that intervened. Because between the time that Griffiths described and published his findings in the late 1920s, early 1930s, it wasn't until the 1940s that additional scientists were able to essentially pick up where Griffiths left off and try to figure out what was the cause of this transformation of heat-killed smooth to living smooth. In the late 1930s, early 1940s, additional scientists followed up on Frederick Griffiths' discovery of transformation and determining what was the transforming principle. In other words, what was inside of a cell that could cause smooth, heat-killed Streptococcus pneumoniae to be transformed into living or growing smooth Streptococcus pneumoniae by simply mixing the heat-killed material with rough, growing Streptococcus pneumoniae. Oswald Avery was the scientist who started to take up the investigation of what was the transforming principle, working with his colleague, Colin MacLeod, in the very early 1940s. Using various techniques to extract all of the bits and pieces of cellular material and simply leave the biochemicals in a solution and performing transformation in a test tube was how Avery and MacLeod began to discover what was the cause of transformation. MacLeod moved on to other research, and then another scientist by the name of Maslin McCarty continued work with Dr. Avery to determine the transformation principle. By taking different mixtures of extracts of smooth Streptococcus pneumoniae and rough Streptococcus pneumoniae and allowing transformation to occur in a test tube, Avery, McCarty, and MacLeod are known now for the discovery of the identity of the transformation principle. Essentially what they were able to do is take extracts of the biochemicals, essentially the proteins and nucleic acids found inside the smooth and the rough strep pneumo, and simply mixing those chemicals in a test tube with various enzymes that would inhibit different kinds of proteins and nucleic acids. When the mixtures and transforming biochemicals were mixed with proteases, enzymes that would essentially inhibit the function or the activity of proteins, transformation still occurred. If you mixed the biochemicals in a test tube and you added RNase, or an enzyme that would inactivate RNA, transformation would still occur. If you mixed the chemicals again into let's say a third test tube or flask, but this time added DNase, or enzymes that would inhibit the function of DNA, then these scientists discovered that transformation did not occur. It led to the distinct conclusion that DNA must be the transforming principle, and that DNA was responsible for the transformation of the living rough Streptococcus pneumoniae when exposed to what I often call the little dead body parts of the smooth, virulent, heat-killed Streptococcus pneumoniae, that it was the DNA in that mixture that was somehow taken up by the rough cells and then transformed those rough cells into smooth. For their discovery, Avery and McCarty and MacLeod have been identified for many years as sort of at the heart or the very beginning of the further and final discussion and discovery that DNA is the source of genes and hereditary material. Oddly enough, prior to the 1940s and 50s, when scientists firmly established that DNA is the source of genes, Many scientists thought that proteins were genes, and that DNA was simply a sort of a dull, monotonous molecule and really wasn't responsible for anything to do with heredity at all. So the contributions by Griffiths, Avery, McCarty, and MacLeod are really at the heart or fundamental to our knowledge in modern genetics today, all using prokaryotic examples like Streptococcus pneumoniae. Now, oddly enough, transformation doesn't naturally occur easily in most bacteria. In fact, in order to deliberately cause transformation in a laboratory today, we often have to treat the bacterial cells with electricity or other kinds of chemicals. It just so happened that Streptococcus pneumoniae is one of the few species that will naturally undergo transformation, which of course was to Griffith's credit and benefit. He didn't know it at the time, but he was quite fortunate that he chose that particular model and that particular organism to study, in large part because he was trying to invent a vaccine, ultimately, to prevent pneumonia in patients. So a long history based on and involving this one little technique and procedure. We now know that if we look more closely at bacterial cells, that the bacterial cells that are able to undergo transformation have to be what we call competent. They have to have certain unique characteristics about their cell wall or the peptidoglycan and their outer surface that other types of prokaryotic bacteria might not have. So here we have a close-up example of how we could possibly transfer pieces of DNA from the environment into a competent cell. Now, Streptococcus pneumoniae will allow this to occur naturally because of some of its unique characteristics of its cell wall or outer covering. But if we want to perform this technique in any bacterium, we can either treat it with chemicals or we can even use electric current, as illustrated on this slide. By treating or exposing the bacterial cells to essentially a little electric current, we can weaken or cause the formation of openings in the peptidoglycan layer that will allow fragments of DNA to pass through not only the peptidoglycan layer but also the cytoplasmic membrane. The little fragments of DNA are illustrated in red on this slide, and even though it's double-stranded DNA from the environment, it's simply a piece of single-stranded DNA that eventually is allowed to pass through. These are very small fragments, and in fact, of all three mechanisms of lateral gene transfer, we're going to find that transformation often allows only the smallest, tiniest pieces of fragments of DNA from the environment to be passed from one bacterial cell to another. Once the single strand is inside the cell, then there are mechanisms and enzymes that at random will splice this newly introduced DNA into the bacterial chromosome. Again, these are all random events, and so where and how the DNA is added and whether or not it's able to be expressed or transcribed and translated, again, is all random. So often we're simply looking for a needle in a haystack, and that it occurs and is successful at all is quite interesting, I think. A second mechanism of gene transfer occurs naturally by some types of bacteriophage that we call temperate bacteriophage or temperophage. Now, we'll come back to these same kinds of examples when we discuss viruses in class, but again, a naturally occurring process for certain types of bacterial viruses is the incorporation of viral DNA into the bacterial chromosome. Now, phage is referring to mouth or eating, and a bacteriophage originally was discovered because we'd mix cultures of bacteria and see that there seemed to be something in the culture that was eating the bacteria. Well, we've now identified whatever this little eating bacteria item was as a bacterial virus. And temperate phages, or temperate bacteriophage, have two alternate growth cycles. One's called the lytic cycle, which is the typical kind of viral infection that we think of when we think of viruses. And then the alternative for prokaryotic bacterial viruses is the lysogenic cycle, or lysogeny. Now, temperate bacteriophage, when they attach to their target bacterial cells, will introduce their genetic material, and this example is going to be a bacterial virus that has DNA as its nucleic acid core. And we can see for step number 2, bacteriophage has introduced its DNA, and the viral DNA, as well as the prokaryotic chromosome, are in little circles, are circularized. And if we want to continue from 2 to 3A in the lytic cycle, we're simply going to have this viral nucleic acid directing the synthesis of new viral proteins and new viral nucleic acids, the assembly of all those components into new viruses, and eventually release from the bacterial cell in a mechanism called cell lysis, or the lytic process. Cell lysis means that the cell is ruptured. We have to bust out or break the cytoplasmic membrane and break the cell wall or the peptidoglycan layer, or destroy it, in order to release the newly assembled and matured viral particles. These newly synthesized virions can then go on and infect new uninfected bacterial cells. And those are the typical steps of the lytic cycle. There are lots of examples of both prokaryotic and eukaryotic viruses that perform this type of infection, or this type of growth cycle. Now, for temperate phage, we have an alternative cycle to the lytic cycle, and that's the lysogenic cycle, or the condition that we call lysogeny. When we get to step 2, where we have the phage DNA in little circles, as well as the prokaryotic chromosome, notice that instead of going through the lytic cycle with the synthesis, assembly, and release of new viruses, the viral DNA becomes integrated or incorporated into the bacterial chromosome. We have 2 different sources of double-stranded DNA that are spliced together. The term that we use to describe 2 different sources of double-stranded DNA that are spliced or fused together to create a new construct is recombinant DNA. So recombinant DNA, 2 different sources of double-stranded DNA spliced together. When we see this occurring in a lysogenic cycle using temperate phage DNA, we call this new construct of the viral DNA a prophage. So the prophage is the viral DNA fused with the bacterial chromosome, and now every time this DNA undergoes replication, transcription, or translation, all of the material is performing that function. So any new genetic material that's found on the prophage that might give the bacterial cell a new adaptation or some type of environmental advantage will be useful for the bacterium. But we simply aren't making new viral particles. We're simply passing the genetic material along from one generation to the next. So the lysogenic bacterium reproduces normally until there are certain kinds of either environmental or other kinds of nutritional cues or activators that will result in the excision of the prophage back to an extrachromosomal piece of DNA in the form of viral DNA, which is the little red ring, outside of the bacterial chromosome, which is the sort of purplish blue larger ring. When we get to that middle step again, that's step number 2 in the center of the image, now we'll find that, again randomly, based on various different kinds of physical or chemical activators or cues, this viral DNA can either enter back into the lytic cycle or go back into the formation of a prophage for the lysogenic cycle. Either way, this process is called transduction, because at step number 5, when we have the excision of the prophage popping back out into a complete construct of DNA that's going to be used by the virus, when that sort of excision occurs, it's not perfect, and we find that there are bits and pieces of bacterial chromosomes that are often attached to the viral DNA. So we have sort of a combination of viral and bacterial DNA that's excised out, and then if this newly sort of recombinant DNA is now packaged inside of a viral capsid that then goes on to infect a different bacterial cell or another bacterial cell when it's assembled and released, then we've essentially modified new bacteria using viruses, bacterial viruses, as our transport device. We can package bacterial genes and pieces of bacterial chromosomes inside the viral capsid. And lysogeny is one way that bacteria can naturally transfer bacterial DNA with the use of bacterial viruses. This is the image of transduction from our microbiology book. Notice that the example of the lysogenic cycle isn't quite as complete as the previous image, but I thought I would include it so you can compare the two images. They're both giving you the same information, that the lysogenic cycle that occurs when we're using a temperate bacteriophage results in the incorporation or the formation of a prophage in the lysogenic cycle. The third mechanism of gene transfer is DNA transfer by conjugation. Now, of all three mechanisms, conjugation has the potential to transfer the greatest amount of DNA, and also can result in an interesting recombinant form of bacterial cell that has an even greater frequency of mixing up genetic material. The transfer of DNA by conjugation is going to rely on the presence of a transfer device called an F. pili, or that would be plural, and the singular is pilus. So an F. pilus would be the singular or one of these transport structures. F. pili would be more than one. Notice in this scanning electron image that an pili is a structure found on the surface of the bacterial cell. It emerges from the cytoplasmic membrane through any cell wall or cell layers on the outside of the membrane as an external structure that can be used for attachment of one bacterial cell to another. If you go back to that chapter where we compare differences between pro and eukaryotic cells, you'll find that one of those structures that's unique to prokaryotes that we can find on some prokaryotic organisms or some prokaryotic cells are structures called pili. They can be used for attachment, or they can be used for exchange of genetic material. And what we simply do is describe which one are we talking about. Are we talking about attachment pili, or in this example, are we talking about conjugation pili? Now here we have an example at the cellular level of how conjugation works. We're going to find that bacterial cells that can produce F. pili have unique genetic material inside called an F. plasmid. The F. plasmid is responsible for the production of the F. pili because there are genes on the F. plasmid that, when transcribed and translated, result in the production of a specific kind of protein called pilin, which is used in most of the structure of pili. So if we can produce pili, we're going to be called an F plus cell. So F plus simply denotes that the cell has the F plasmid inside and is capable of producing F. pili. or conjugation pili. Bacterial cells that don't have these plasmids are identified as F-. Now, when we have the F. pili on the surface of the F+ cells, this pili can serve as a conjugation bridge or connection between the F+ cell and the F- cell. So if you take a look on the left-hand side, you can clearly see the middle image shows essentially the attachment of the F+ cell to the F-, and notice what's happening along the conjugation bridge. A copy of the F plasmid is being transferred from the F+ cell to the F- cell. This is conjugation, the transfer of large sections or pieces of DNA from an F+ to an F- via the conjugation bridge or the F pili. Now eventually a whole complete copy is transferred, and now the F- cell is no longer F- because it has its own plasmid copy, which eventually is completed and made in the form of double-stranded DNA. And now that F- cell has been changed to an F+. In 1946, conjugation was discovered by the microbiologist Joshua Lederberg and the geneticist Edward Tatum. They used different strains of E. coli that had different nutritional requirements. And in fact, the discovery that bacteria could exchange genetic material through direct contact, or what, for all intents and purposes, often looked like sexual reproduction, because we have genetic material from two different sources that are transferred by direct contact from one to another, was quite a landmark discovery. Before that, it wasn't even conceived or thought that bacteria could touch one another and transfer genetic material. There are a variety of different kinds of plasmids. F plasmids are not the only types of plasmids that have been discovered. There are a whole variety of plasmids that have different kinds of genetic material incorporated into their structures. And one specific example is the resistance plasmid, often called an R plasmid. Resistance plasmids have genes for antibiotic or drug resistance. These are naturally occurring. Realize that antibiotics, the classic definition of that word, antibiotics are chemicals produced by fungi to control or kill bacteria. It's a form of chemical warfare that occurs between these two general categories of microbes. The fungi try to keep the bacteria from growing so that they can essentially grow in some defined biological habitat. Now, the bacteria fight back chemically, producing antibiotic resistance chemicals or resistant substances. Often these are enzymes that will break down the antibiotic, or they could be different kinds of proteins that prevent the antibiotic from functioning properly. In any case, resistance plasmids can also be transferred by conjugation. Realize that cells that can conjugate are still going to have F plasmids in addition to R plasmids. And it's essentially the transfer of resistance plasmids from one bacterial cell to another that have resulted in the huge number of antibiotic-resistant bacteria that we now find that can cause infections today. And the fact that plasmids can be transferred not only by conjugation, but we find that these can also be transferred by transduction and even transformation, has allowed different bacterial species to quickly transfer drug resistance from one bacterium to another.